To try to understand why we came up with these goals, I thought it important to review the history we had with Aldurazyme. It's a story of success and yet tragedy. Success that we ultimately got development, but tragedy that we encountered so many delays that it ended up making it very difficult for many patients. We did an original 10-patient study, which was very successful, using markers of storage, which are a way of looking at how the disease is being treated. And we had positive canine data showing how that related to the disease pathology. It was very similar to what was done for Gaucher disease. The study came out with very positive results, both showing improvements in liver size as well as the urine sugar reduction. And we also found a number of other clinical benefits. But the FDA felt uncomfortable with those surrogate endpoints because they'd never been used before in a clinical or drug approval. And therefore, they felt that we needed more validating human data. Data we don't have because this was the first clinical study. So there is the problem. How can we prove endpoints are meaningful when studies have never been done before? It's a fundamental problem for rare diseases. Aldurazyme was eventually approved, as you all know, and there have been a number of patients on drug for many years now. But with the urine GAG endpoint rejected by FDA, and then an additional delay related to statistical issues and our need for extension data to help convince the FDA of the efficacy of Aldurazyme, we ended up needing an advisory committee meeting in which outside experts come to the agency and advise on whether they think the drug was effective. Now, they voted 12-0 the drug was effective, and the FDA then approved the drug in April of 2003. So ultimately a good outcome, but look at what happened. If we had gotten approved based on our surrogate endpoints, we would have been available in year 2000. And if we had not had the statistical issues come up with what are the best way to analyze data, we would have probably gotten approved by 2002. Instead, we ended up advising committee 2003, approval in 2003, in April 2003. So that's a 3-year delay. But worse than the 3-year delay, in fact, was that other programs got canceled, including the MPS 7 program and a Morcure program that was ongoing at the time. Now, Matthew is a kid who has MPS 7, and his disease has been treated since 1993 in mice, but humans haven't been treated. We think that's a tragedy that should be fixed. And that's true for a lot of very rare disorders, as well as those that might affect difficult tissues like the brain, like Sanfilippo. And we think those things need to be changed so that these diseases can get treated when the science is there. If you look at the system itself, what you're finding is that, in fact, among ultra-rare disorders like these biochemical disorders, that affect maybe less than 6,000 in the US, relatively rare everywhere, those disorders, in fact, have only had maybe 2 or 3 products approved for the last 12 years. Even though there have been many more designations coming out, meaning people are trying to get drugs developed, but the amount of drugs getting through hasn't really changed in all those years, and I think that's a problem. We need more than 2 or 3 a year when we have several thousand that are untreated today. It will take us many decades to get to treatment, even if we have the science available. One of the challenges, of course, relates to how you determine if a drug works. And the accelerated approval regulations were established to help get cancer and AIDS drugs approved. And what it said was these were terrible diseases, and you ought to be able to use a urine test or a blood test if it was reasonably likely to predict clinical benefit. But what's really happened is that cancer and HIV or AIDS have benefited from the accelerated approval regulations, but the genetic diseases and the biochemical genetic diseases, disorders like MPS or ML, haven't gotten the benefit of that regulation. Only one genetic disease has been approved based on accelerated approval. That's despite the fact that these rare biochemical disorders probably have better markers than almost any of the other disorders they're talking about. We think this is a shortage in understanding at the FDA, understanding of the regulatory science, and we think this could be improved. So the goals for the Cure the Process campaign are try to get the right people in place by establishing a specialized review division within the FDA. These people would be trained and knowledgeable about biochemical and genetic disorders. We want to improve the accessibility of the accelerated approval process. That is, provide some way of determining how to use a surrogate endpoint and to be able to get more rare disease drugs access to the accelerated approval pathway. And finally, we want alternative, more powerful study designs and analyses. This should be obvious that we can use them, but we need agreement with the agency of what type of study designs are possible and usable. Those improvements, we believe, would have a substantial impact in improving the pathway for development of rare disease therapeutics. Now, to try to improve the FDA's review of these drugs, we're proposing that a specialized review division be established within the FDA, within the CDER, or the Center for Drug Evaluation and Research. That group within the FDA reviews the majority of the drugs and protein therapeutics. And that group needs to have more specialization in our disease areas in order to understand how to regulate them. You'd expect people to be trained in the disease area and have seen patients with those diseases so that they know how to make sense of clinical data and how to make sense of surrogate endpoints. We'd want those people to have more time to be able to manage those complex disorder applications, as well as time to connect with NIH and be up to date with the latest science and the latest information about that disease and its treatment. Those kind of important joint appointments and activities we think would maintain a highly knowledgeable and effective FDA review group. And we think this is an important part of making that first step toward improved drug regulation for rare disorders.